Yeah, thank you for the introduction. Last talk for today. Let's briefly talk about NanoAIRR. So, sequencing of B cell and T cell receptors is important to better understand their function and identity in disease context. Key aspects are to look into diversity, repertoire diversity, lineage evolution of clonal groups, and also their spatial context within the tissue. Established workflows mostly focus on short-read sequencing technologies. However, long-read sequencing technologies offer full-length receptor resolution, and with that, a more accurate repertoire representation. I want to introduce you to a new set of methods that we are developing, the NanoAIRR methods, utilizing Nanopore long-read sequencing for B and T cell receptor repertoire sequencing on both bulk and 10x Visium spatial resolution, with read length of up to 3.5K base pairs. These experimental methods offer full-length receptor resolution, so no pairing or assembly steps are necessary. In addition, we are developing the NanoAIRR computational bioinformatic toolbox, currently implementing 6 core functionalities and combining existing software, for example, from the incantation framework, with new functionalities to accurately process and annotate your receptor sequences that you generated with Nanopore sequencing. NanoAIRR offers sequencing error correction through UMI grouping and consensus building, and also subsampling of very large UMI groups to efficiently process your data. Let me show you how well it works. So as a technical validation, we compared Nanopore reads and PacBio reads, both from the same sample, both processed with NanoAIRR. You can see the very comparable step-by-step retention of reads through the different NanoAIRR steps. And the final yield of productive receptor sequences is basically similar. However, Nanopore sequencing is more accessible in the labs and more cost-effective. Spatial plots from initial data analysis show that the receptor sequences that we are able to capture and process, so here in red and blue, associate with regions of interest. In our case, these are immune cell aggregates within solid tumors, which confirms a proof of concept that with NanoAIRR we are able to capture and extract meaningful and spatially resolved clonal information from complex tissue. With that being said, I would like to thank everybody involved in this work, specifically my supervisor Katharina Imkeller and Samira. Thanks to the organizers, and thanks for listening.
In case there are any quick questions for the short talks, if not, we'll move to closing remarks. There's one, and then we'll move to closing remarks.
Yes, thank you for the great talk. You working? Okay, so I'm just wondering if you tested the NanoAIRR on R9 or R10?
R9 or R10, can you see which one?
The newest one, R10.
So I'm just wondering if it was for like R9, which has a higher error rate. Do you think so?
What was the last part? Sorry.
Do you think it was for like R9, which has a higher error rate? No.
We didn't do this for R9, no.
Do you think it would work? Would it still work?
It would work, yeah. The input is probably the same. We use also the official Nanopore base caller. So the old technology probably has the same output, which you could feed into the tool.
Okay, thank you. So thank you everyone for coming. I think this was the first